So can you just say who you are and where are you from?
Okay. My name is Omar Rodriguez. I am from Puno, Peru, and I have studied biologist. So now I am a biologist.
Fantastic. And so why did you decide to take this course?
Well, I'm very interested in evolutionary biology. So I didn't have a background in genetics, but I understand that genetics is very important to better understand evolution and also to can research about this. So I see that I have this opportunity and I take it. Yeah.
And so what was your level of background coming into this course? This was called, like a genetics or genomics course. Had you had those types of skills before?
No, I didn't have I have to say any skill in genetics, but I was reading a lot before coming about the basic genetics and terms like DNA and the genome or this kind of thing. So yeah, I have something. But
And so you wanted to come here to get more training in the techniques, is that right?
yeah, exactly. I didn't have any background and I didn't work never in a laboratory doing this kind of things. So yeah, that's what I was looking.
Are there lots of opportunities to work in labs like this and do genetic work?
You mean here in Peru? Well, no, I don't know about any opportunity like that. So no, I think no, there is no.
And so where you received your degree, are there not so many opportunities to work in a laboratory and do these types of genetic experiments?
No, there were no opportunities for me. Yeah, but also working in this kind of things of genetics and this is like something recently that I decided recently. Before this, I consider myself a field biologist, so I try to be most of the time in the field, and I used to work or try to work with wildlife. So I didn't search too much for these kind of things. So that's why maybe.
And so what did you first think when you first heard about this course?
Well, maybe it's important to first tell how I find this course. I find the page of FPI, and they were offering these courses, but not this exactly. They were offering one about birds. So I applied for a scholarship in this course, and I won. But the course didn't open because there were not enough people. So they tell me, okay, you can wait until the next course, so you can go there. So yeah, the time comes of the new courses, and I find that they open this course. So I decided to change because I was very interested, as I told you, in learn more about evolution and genetics. So yeah, I just say, please, can you move me to this other course? And they say, we're gonna check if your application is good enough. And yeah, hopefully it was.
Fantastic. And so coming in with not so much experience doing things like, could you tell us a little bit about the types of techniques you've done so far, even the basics, like, had you done pipetting, had you done, running gels, sequencing? Can you describe the types of experiments that you've been able to do for the first time?
Here, you mean?
Mm-hmm.
Okay. Well, yeah, everything was new. So, first thing, pipetting, was a completely new thing for me. I have done in my university pipetting, but with other kind of pipettes. I think are made of glass or something like that. Also run a gel. Before coming, I saw some videos about how to run a gel or about how the electrophoresis works, but I didn't have even an idea of the size of a gel or anything like that. So then I come and I realized that was something so small. I don't know why I imagined that it was a very big thing. Then, well, using the quantus, for example, was something that I don't even imagine that can exist. Something that can tell you the quantity of DNA that you have in a tube. So yeah, I think everything was new. Yeah.
And then what did you think about, those are some things that have been around for a long time, right? These types of techniques, but restricted to wet laboratories. And then what did you think about the ability to actually sequence the DNA with a portable sequencer?
Well, I think it opened a new world of opportunities for people who wants to do this kind of things in a place like this, where you don't have access maybe to a whole laboratory with all the equipment that you normally have in for example, in a university. And it also, I think it also brings more, like, how can I say it? I don't know. Like more topics to studying, things that you can probably not do if you can't read your samples in real time or something like that.
Fantastic. And now that you've gone through all of these steps, do you feel like you're more prepared to do future experiments?
Yep, I'm sure I can. I also have some ideas that I would like to apply with the new techniques that I recently learned. I also know that I need more training, but hopefully in these future years I will be ready for maybe publish some things and maybe study more, maybe in a master or something like that.
Fantastic. So if you were to sort of, summarize the last 2 weeks almost, what would you say you did here, and why would you say it's important?
Okay. Well, first, these weeks were weeks of a lot of learning. We do first learn about the laboratories, all the things that I just mentioned before, all of this pipetting and electrophoresis and all of these things. Also, well, then we start doing this of the barcode, with which we want to find the genetic code of a species, so we can recognize a species and differentiate it from each other. How can I say it? We take samples of about 100 species between plants, insects, ectoparasites, monkeys, bats, rodents, I think. A lot of organisms, different organisms. Then we have to prepare a lot of things to finally end with a small tube with all the samples inside. It was very, very exciting and very funny to see all this work all together in a small tube. Yeah, and then we use finally the MinION, and we are now having in real time all the results of our sequencing. So we have already found some species, and we hopefully are going to finally have our tree, that phylogenetic tree that I was very excited to do.
Awesome. And so sort of concluding, in addition to the training, in addition to learning the techniques, why do you think it's potentially important to do this within either this area or in Peru rather than sending the samples internationally? Why is that potentially important?
Well, I think it has many applications and it can be important in many cases. But for example, one case that we have here, is with one species of monkey that we are working with, that is the howler monkey. That in a rescue center they want to reintroduce this species into the wild, but they can't because they don't know if the species is the same that the native here. So if they just put in free species that is not from here, it can become a problem. Well, also, as I say before, we have like more opportunities to do research in the field, and not only here but all over the world. Everybody can have ion in the pocket, I think, and all these other tools that we use, like the mini PCRs and these kind of things. So, well, yeah, I think.
Awesome. No, that sounds great. And it might sound a little repetitive, but can you say your name, where you're from, and what you do one more time?
Okay, sure. My name is Omar Rodriguez. I am from Puno, Peru. Sorry, what was the last question?
That's okay. And you can say like I'm a biologist, but can you start from the beginning?
Okay, okay.
Uh-huh.
My name is Omar Rodriguez. I am from Puno, Peru, and I am a biologist.
Awesome.